Mendelian Genetics. We are going to use Gregor Mendel's laws. Those are the laws of segregation and independent assortment to discuss the role of meiosis and define problems, perhaps, or even ask questions about how these things occur and why things turn out the way that they do. Now, dihybrid crosses we use when we try to find the possibility of genotypes for offsprings when we're considering like two traits at the same time. Sometimes this will be if one gene relies on the other, or maybe we just want to have short plants with white flowers, for example. And we want to know, it's easier to not have all of these offspring and over again. If we can do this multiple genes at once, we can make the generation time quicker. So an example for number one would be if we want to cross two tomato plants that are both heterozygous for being tall. So hetero means that they're different. There's our genotype. Red, which means it is heterozygous for red. Note that the recessive traits in tomatoes are short and yellow. So our heterozygous tomato plant has one dominant trait and one recessive trait for tallness, and one dominant and one recessive trait for being red. I didn't realize that tomatoes, yellow was actually recessive. I thought it was just a different kind of tomato, but what do I know about tomatoes? Okay, so write the genotype of the parents. This problem, it says that they are both that. So mom is big T, big R, little r, and dad is big T, big R, little r. All right, sort the alleles and ask yourself what combinations could you see. Now, before we do our Punnett square, I want you to think of something on how we're going to write this. If mom is heterozygous for two traits, and dad, the dead tomato plant, which is weird to think of, but is also heterozygous, remember that things independently assort. So this T can be paired up with that R, or this T can be paired up with that R. And then this T can be paired up with that R, and then this T can be paired up with that R. And that's the same thing is going to happen for dad. We have to put together the combinations of how they would independently assort. So if I am big T, big R, little r, then my big T can go with the big R, or my big T can go with the little r. And if I'm the little T, my little T can go with the big R, or my little T can go with the little r. And so that's what it means by they're independently assorting. Now it says that both mom and dad here are the same, right? They're both heterozygous. So we'll just fill that in for both parental generations. And now we're just going to carry them all the way down. So I'm going to take this big T right here, put it all the way down. And this big R, put it all the way down. All right, and then this big T, I'm going to put it all the way across. And this big R, all the way across. And now we're going to fill in the whole entire box. Alrighty, so you might want to pause the video here for a second and fill in your dihybrid cross. You want to write the combinations on the side of the Punnett square, and in this case, we want to use the same combinations at the top side as the other side, because mother and father are the exact same genotype. And then we want to kind of combine these alleles and independently assort them, as explained. Also, remember, both parents are tall and red. So let's find out what the probability is that the offspring will be tall versus short. If I look at my Punnett square, everybody in this shaded area right here is going to be tall. They're all going to be tall because everybody has at least one dominant allele. The offspring in the green shaded area have the probability of being short because they all are homozygous recessive for short. See little t, little t. So our phenotype for tall is 75%, and for short, it is 25%. Now let's do who's red and who's yellow. In order to be red, you have to have at least one dominant R allele. Everyone I'm going to shade like this is going to be red because they have at least one dominant R. And everyone who is not going to be red, who receives homozygous recessive for little r, is going to be yellow. So everybody right here is going to be yellow. In order to be yellow, they had to have two little r's. And so what we care about here is our phenotype ratio. We want to know who's tall and red and tall and yellow, and who's short and red and short and yellow. So let's circle everybody who's tall and red. Right? That's 9. And let's put a box around everyone who is tall but white. Just kidding, tall but yellow. There's tall and yellow, there's tall and yellow, there's tall and yellow. So there's a 3. Now, who's short and red? Short and red, short and red, short and red. 3. And so here we have short and yellow. So this is a pretty typical ratio. In a dihybrid cross, when we have two heterozygous parents, we get a 9:3 to 3:1 ratio of the offspring. And it's possible that they could have all short and yellow offspring, but the chances are that in a controlled dihybrid cross, especially when it comes to plants, that we're going to see this ratio pop up in a dihybrid cross of two heterozygous parents. But these are the allele combinations that we could see and the phenotype ratio that they can express. Now let's have you practice. An example number 2, in peas, purple flowers are dominant to white flowers, and tall plants are dominant to short plants. So a white and short plant is crossed. So let's think about this for a minute. A white plant is recessive. I'm going to use little r's for the white plant, and little t for the short plant because it's recessive, and it's going to be crossed with a plant that is heterozygous for both traits. So the other one's going to be heterozygous, big R, little r, big T, little t. So let's sort the alleles. I think this first one's going to be easy, right? Because every way you sort this, you're going to get the same combination because he's homozygous recessive for both traits. And for the other parent, for the father, remember we're going to have the big R is going to go with that big T, and then it's going to go with that little t, and then the little r is going to go with that big T, and then with that little t. Alrighty, so now we want to write these combinations on the sides and then combine the alleles. So let's fill in the chart. Okay, so I'm going to carry, did you catch my mistake there? I was just writing away, talking and writing. So I fixed it. So I'm going to carry the little r down, R, this little r down, this little r down, this little r, and then this little t, just like r's and t's because they're distinguishable between the capital and the lowercase letter. Sometimes I see a lot of C's and W's, and those can be really hard to tell the difference if it's dominant or recessive or not. Okay, so there's the mother. Now here we go. Here's the father. Get this big R. And then the little r from the father. Okay, now the T. I like to write them big T, little t, because that's just proper biology, proper genetics. Well, how about that? I screwed that up again. So I'm going to just erase these really quickly. Oops, my bad. And we're going to put our little t all the way across here. And our big T goes over here. I often leave the mistakes that I make in the videos because I want you to see that I have to pay close attention too. And it's always good to double check yourself no matter how long that you've been doing this. Okay, so there is our Punnett square. Let's write our combinations and our ratios. So the first I'm going to look for is purple versus white flowers. That is my R. Everybody who has at least one capital R is going to be purple. So everybody right here is going to be purple. All right, and everyone below here is going to be white. Now, who is going to be tall? Everybody but with at least one, this one right here and this one right here, capital T is going to be tall. So we have tall right here and tall right here. This is a pretty unique ratio, isn't it? Because when we have purple and purple flowers that are also tall, we have, we'll say a 4. Purple flowers that are short, we'll have these guys right here. That's also 4. And white flowers that are tall, which are going to be these guys right here. And then white flowers that are short is everybody on this bottom row. So for this dihybrid cross, we have a 4, 2, 4, 2, 4, 2, 4 ratio or possible outcome for these offspring. This is really a 1 to 1 to 1 to 1 ratio if you think about it. This is called a diallelic combination or diallelic ratio. In genetics, we call it a diallelic combination. It's not one of those words that you have to know. It's not on your vocabulary. It just means that all 4 possibilities are going to occur with equal probability. So all the offspring have an equal probability of either being purple or white or tall or short or a combination of the two.